It's important for us to be very clear in the language that we use when we're discussing infectious disease, because some of these terms can be mistaken for each other. For example, it's possible for a person to be exposed to a pathogen but not become infected with that pathogen. It's possible for a person to be infected with a pathogen but not develop outward signs of disease. So the question is, what determines when an infection occurs and when an infection becomes disease? Things like the infective dose plays a role, previous exposure to the pathogen, and whether or not the person has any immune memory to that pathogen, the general health of the immune system in that person, and of course the virulence of the pathogen involved. Let's start first with the idea of infectious dose. There are two concepts we need to be aware of. One is infectious dose, and the other is known as lethal dose. And you'll notice the abbreviations include the number 50. ID50 or infectious dose 50 and LD50 or lethal dose 50 are terms that we associate with infectious disease. So an ID50 describes the number of pathogenic microbes or viral particles, if it's a virus, at which 50% of the exposed host organisms will become infected. So there is a threshold, there's a minimum number of microbial cells or viral particles that the host organism needs to get exposed to in order to become infected, in order to have that event where the pathogen penetrates and starts to multiply. The LD50 describes the number of pathogenic microbes or viral particles at which 50% of the infected hosts will then die. So it's very important for us to understand that different pathogens, because of factors like virulence, have a different infectious dose 50 and LD or lethal dose 50. For example, an organism like Mycobacterium tuberculosis, which is a type of bacteria that causes the disease known as tuberculosis, has an ID50 of 10. You need 10 individual bacterial cells to infect 50% of the individuals that get exposed. Now, Mycobacterium tuberculosis is considered a highly infectious, highly virulent organism, so it makes sense that a relatively contagious organism would have such a low ID50. Compare that to an organism like Vibrio cholerae, which is the causative agent for the disease cholera. That organism has an ID50 of 1,000 cells. Yes, cholera is also contagious, but it's less contagious than tuberculosis. It's virulent, but it's less virulent than the tuberculosis organism. So virulence, again, is a measure of how pathogenic, how virulent an organism is. And again, we can investigate the virulence of an organism by looking for products or molecules that microbe produces that help make it more pathogenic, more virulent. These are often proteins or other cellular molecules. There are virulence factors, in fact, that are part of external structures outside of the cell wall of a bacterial cell. Things like capsules, what are called adhesins. that contribute to the virulence of a pathogen. Some microbes can even secrete toxic molecules that make them more virulent. What a virulence factor does for a pathogen is it allows it to overcome or evade the immune system of the host. It helps it better replicate itself, better multiply and increase its cell number. It helps it sometimes cause damage to the organism. Think of that as helping it to invade tissue. These virulence factors, in other words, give that microbe a certain amount of defensive ability to help it escape destruction by the immune system, and also offensive ability, the ability to do whatever it needs to do inside the body of the host in order to survive. So let's think about a couple of these virulence factors. We talked about the capsule as being a type of structure that we find in some bacteria that exists outside of the cell wall. If you recall, a capsule is a form of the glycocalyx. It's composed of protein and sometimes a little bit of carbohydrate, and it's a very tightly organized structure. Essentially, what the capsule does for the microbe is it helps it evade the immune system of the host. If you look at the image at the bottom of the slide, you can see some rod-shaped bacterial cells. This happens to be Bacillus anthracis. And you can see that although the tissue in the background is staining pinkish purple and the rod-shaped cells are staining blue, there's this halo of unstained area around the bacterial cells. That's where the capsule is. Organisms like Bacillus anthracis, which causes anthrax, Staphylococcus aureus, the strains that cause flesh-eating disease, and some strains of Streptococcus pneumoniae. These pathogens all have these capsules around them that, again, help them in their virulence because they help them stay alive longer in the host by evading the immune system. Adhesins are a virulence factor that actually sort of protrudes off of the surface of the cell. Now, we also talked about some of these adhesins, at least, when we talked about cell structure. Pili, if you recall, help a cell attach to a surface. Pili are a type of adhesin. But there are other adhesins that we haven't talked about yet. Some pathogens actually have structures protruding off their surface that form little hooks at the end. There are spike structures that come off of the surface of certain microbes. Some microbes even have what look like little suction cups that come off of them. I'm talking about some of the single-celled eukaryotic microbes there. Adhesins, again, are going to allow that organism to have a higher virulence because it's going to allow it to stay in the host where it wants to stay, where it's going to have the most success in multiplying and surviving. For example, the organism that causes the disease known as gonorrhea, and that organism is called Neisseria gonorrhoeae. That organism has a preference for the urogenital tract. And as you can imagine, it's difficult for a bacterial cell to stay in the urogenital tract because it tends to be flushed out with the urine. So it actually has adhesins that come off of the surface of the cell that help it bind to the epithelial membranes inside that tract. And it allows the organism to stay put and multiply and cause disease. So on this slide, we can see some of these adhesion type structures that we see in microbes. Up at the top, for example, you've got a drawing of the human immunodeficiency virus. This happens to be one of the viruses that have these very distinctive spikes coming off of the surface. And remember that it's these spikes, these proteins that stick off of a viral particle that allow it to bind to receptors on the host cell. Down at the bottom, we have a drawing of two Giardia organisms. And Giardia, if you recall, this is a pathogen that people tend to contract in contaminated water. I'm thinking about, for example, water that comes out of streams and rivers that people don't disinfect. Giardia can cause a pretty miserable gastrointestinal disease. And if you look at the bottom surface of this organism, it's got this disc that actually functions like a suction cup. It allows the Giardia organism to literally suction itself to regions of the intestine where it can stay and multiply and not just get passed out with the fecal material. Up here in the middle, you're looking at a microbe that has both flagella and little hooks off of the end of those flagella that help it attach itself to surfaces. Over here, this is actually a treponema organism, but I think you can just see it's got a hook at the end of it that's going to allow it to attach itself again to the surface of tissue. These are all adhesins. When we talk about virulence factors, we also have to think about things, molecules that the microbe is secreting. There is a type of enzyme that we've discussed in lab called an exoenzyme. This is an enzyme that a microbe actually secretes or releases from the cell, and the enzyme is capable of digesting materials that are out in the environment. There are times when an organism uses exoenzymes to digest exterior molecules so that those molecules can be absorbed as nutrients. But there are also times when exoenzymes can be used by a pathogen to destroy tissue that's outside of the microbial cell. Some of these exoenzymes include things like collagenase and homolysin. Homolysin is a virulence factor that, like the name suggests, hemolyzes or breaks apart red blood cells. Collagenase, as the name suggests, is an enzyme that can digest away collagen. In terms of toxins that can be secreted by pathogens, there are what are called exotoxins and endotoxins. Exotoxins are produced by the pathogen and then purposefully secreted. Endotoxins are different. We've mentioned this term before in earlier lectures. An endotoxin is simply a structural component of the pathogen, a normal structural component of the pathogen. If you recall, we referred to gram-negative bacteria having lipopolysaccharide out on the outer surface of their second membrane. And lipopolysaccharide, or LPS, is a classic endotoxin. Endotoxin becomes problematic for the host organism when large numbers of the pathogenic microbe die and start to degenerate. That's when endotoxin becomes toxic to the host. Exotoxins are different. Exotoxins are specifically produced and secreted by the living cell. So these include things like various neurotoxins. Think about tetanus toxin here or botulinum toxin. There are cytotoxins, molecules that are just toxic to cells in general. Pertussis toxin falls into that category. And finally, there are what are known as enterotoxins. These are toxins that specifically affect the gastrointestinal system. Cholera toxin is a classic example of an enterotoxin.